to help you guys a lot for when you guys are taking the exam. Some things to consider when you guys are taking the exam: you have a long time to do it, right? So instead of the regular exams that you guys are used to having, you guys can take your time and really think through the answers. So really go through them and make sure that what you are reading is what you're thinking, like, is what's being said. Because I know sometimes when you guys go through it, if you rush through it, you can misread it, and one word, as you guys know, can change the answer completely. So starting with the first one, which of the following is true? What do you guys think for A? Humoral immunity is mediated by antibodies. Yeah, that is true. Good.
What's
Humoral is just a term for the immunity that's basically, like it says, just mediated by antibodies. So what types of cells produce antibodies? So is it T and B, or is it just one of them? So B cells are going to be what secrete antibodies. But you guys remember that there is a type of T cell that can stimulate the B cells to secrete their antibodies, right? Okay. So B, cellular immunity is mediated by antibodies. So we just said that antibodies was for humoral, so what is cellular for? Good. So then C, humoral and cellular immunity are part of the adaptive immunity. Good. Okay, so with regards to the innate immunity, cells involved include macrophages. Okay, good, that's true. So if you guys are confused as to what's an innate versus what's an adaptive, what are the only two that are in adaptive? Say that again? T and B cells, good. So if it's not a T cell or a B cell, it's going to be involved in innate. So B, it does not need induction and is the natural resistance our body has towards pathogens. Good, that is true. Mechanical barriers include the interferons and the fatty acids on our skin. So this one is false. What's false about the sentence? What types of barriers are these? The interferons and fatty acids. They're not mechanical, but they're good, chemical. And then D, chemical factors are not a part of the innate immunity. That's false, good, because they are a part. Okay. So number 3, which of the following is true? Animals lacking innate immunity cannot clear pathogens, and the pathogens' numbers increase rapidly. Good, that is true. So if you were looking at the plot, I know she gave you guys in your PowerPoint slides, this would be time on the bottom, and the number of cells, the number of pathogenic cells. So if you are lacking an innate immunity, what's the line going to look like? Right, it's going to go straight up. Yeah, really severe slope going up, but that's a bad picture that I drew, but we'll see the other ones and it'll make sense. So good, A is true. What about B? Animals lacking adaptive immunity do clear pathogens somewhat, and the numbers continue to increase at the same rate as animals that do not lack an adaptive immunity. Good, right. So if they're lacking an adaptive immunity, they're going to clear the pathogens somewhat because they have an innate that's going to clear it somewhat, right? But the numbers are going to increase not at the same rate as animals that do not lack an adaptive, but they're going to increase more than that because they don't have an adaptive to help them out. So this one on the line is going to look more so like this. So it's not as severe as if you're lacking an innate, but they are going to continue to increase. So I'll put in here, this is no innate. It's no adaptive. So then C, animals with both an innate and an adaptive immunity increase the rate of microbial growth for a period of time, but then decrease with the result of final clearing of infection. Good, that's true. So the pathogen's going to increase for a little bit, but then it's going to decrease and eventually get cleared. Okay, so D, animals lacking an innate immunity and an adaptive immunity will not live a long life due to the number of pathogens and diseases the animal is subjected to. Yeah, that's true. If you don't have an innate and you don't have an adaptive, you basically have no way to clear pathogens. So once you get infected by something, you're kind of SOL. Okay. So for B, animals lacking adaptive, they are going to clear them somewhat because the innate's going to clear them out some, like just a little bit, but overall, it's not going to be enough and they're going to continue. Okay, so which of the following is false? Innate immunity is nonspecific. Good, that's true. Adaptive immunity is specific. Good. The mucous membrane is much more susceptible to penetration than the unbroken skin. What do we think? Yes, that is true. And tears contain lysozyme, which is a chemical factor in adaptive immunity. Why is this false? Louder and prouder. Good, because it's in the innate immunity, not adaptive. Okay, so gram-positive bacteria is inhibited by bile salts. Good, that's true. Is attacked by fatty acids. Good. This one's actually going to be for what? Gram-negative, good. Okay, and then lysozyme in tears and saliva are more effective against gram-positive. Good. Okay, so which of the following is incorrectly matched? Phagocytins, chemical factors, and blood proteins. Yes, that's going to be true. What about lysozyme, chemical factor, and blood protein? Good, that one's also true. What about normal flora and microbial factors? Good. And phagocytic cells and microbial factors. Why is this false? What is it supposed to be? Good. Those are going to be phagocytic cells that you're thinking of, and those are specifically going to be the cellular factors. The phagocytins are going to be like chemical factors and blood proteins. That's like where they're classified under, because they're not necessarily like cell factors. They're mostly like chemicals. Okay. Which of the following is true regarding phagocytes? And if you guys have just came in, where you guys can find this is if you go to ucfstudyunion.wordpress.com and ignore my handwriting on this because it was terrible, and go under the biology tab. That's where you guys should be able to find the review for this. Okay, so number 7, which of the following is true regarding phagocytes? They are part of the innate immunity. Good. Their primary function is to engulf and destroy pathogens. Good. They can also act as antigen-presenting cells and generate a peptide antigen that will activate specific immune responses. Good. There are 3 main types, including neutrophils, monocytes, and macrophages. Good, so all of them are true. Okay, so which type of phagocytic cells contain a segmented nucleus with granular cytoplasm that has motile granulocytes with large numbers of lysosomes? Good, it is neutrophils. So 9, monocytes circulate in the blood until they are attracted to the site of infection, in which they change into macrophages. Good, that is true. Okay, which of the following is true? Complement inhibits phagocytosis. Good. And where is complement found? Is that going to be an innate or an adaptive? Yeah, it's going to be in the innate. Is complement then going to be specific for the type of pathogen that's inside, or is it going to be, are they using the same complement pathway regardless of what it is? Good, they're going to be using the same one regardless. So it's non-specific. Okay, most phagocytic processes are enhanced 10 times by C3-C3R interactions. It is 100 times good. And which one would have the C3 on it? Is that going to be our cells, or is that going to be the pathogen? Good, so the C3 is what our cells are going to place on the pathogen. And then where would the C3R be? Good, on our cells, right? Because the C3R is going to be the receptor for the C3 that we place on the pathogen. Okay, one of the primary roles of complement in phagocytosis is opsonization. Good. And what does opsonization mean? Good, yeah, it's basically placing this tag, the C3 tag, on the pathogen so that our cells know to destroy it. Okay, so which type of bacterium has carotenoids which quench singlet oxygen and prevent killing? Staph aureus, Mycobacterium tuberculosis, I hate saying that, I'm sorry. Strep pneumonia? Good. So tell me about Mycobacterium TB, what happens with that? Good, exactly. So the cell wall for Mycobacterium, they're actually going to use their glycolipids. So it uses glycolipids. And that's going to absorb what type of radicals? Hydroxy radicals, good. And what types of anions? good, superoxide. And what about strep pneumonia? Okay, good. So what do they use the capsule for? Yeah, good, basically. So the capsulated ones are virulent, and the capsule basically helps them in multiple different ways, and one of them is that it makes it very resistant to being phagocytosed. Okay. Which of the following is paired incorrectly with regard to the three functions of phagocytes? A, phagocytosis and engulfment. That's good. Okay. What about B, pathogen destruction and reactive nitrogen? False. Good. So what should it be? Reactive oxygen. Good. And then C, antigen processing, target cell lysis, and cause inflammation. Is that true or false? True. Good. Okay, which is placed in the correct order of antigen processing? Presentation of Th2 cell to activate T cells, then plasma cells, then activated T lymphocytes. What do you guys think? True or false? False. Good. B. Presentation of Th2 cell to activate B cells, then release antibody, then form plasma cells. So this one, okay, so who says true? Okay, now no one wants to answer that. Okay, it's fine. Yeah, it is false. Why is it false? Yes, good. It's going to be Th2 to activate the B cells, then the B cells mature into plasma cells, and then those plasma cells can secrete the antibodies. Good. And why do you guys think that they have to mature into plasma cells first? Say again? Okay, yes, for memory. And so basically, like, if we were all B cells, all of us are B cells right now, and a pathogen came in, only one of us can technically secrete antibodies for that pathogen. So instead of having us all secrete antibodies at the same time, basically the one that could, so if it's me, it's like, oh, I got this is my job, I'm going over there. I then mature into a plasma cell and secrete my antibodies, and you guys don't have to, basically. So it's saving the B cells from overworking themselves and like causing all of these antibodies to be secreted for no reason. Okay. Yeah, that's why it's specific too, because all of us would have the same, what region on our antibody would be the same if we were all B cells? Yes, someone said it. No? What region on the antibody is the same for every single B cell? For every single antibody, I mean. Not the part that holds the antibody. So you have the constant region and you have the variable region. What region binds to the what region binds to the antigen? It's going to be the variable part. So all of the antibodies that you have are going to have the same constant region. It's constant throughout your antibodies. And then the variable region is going to determine which antibody can actually go and attack that pathogen. So if someone, like I said, a pathogen came in, we all have different, basically, molecules in our variable region. Only one of us can bind with that. But we're all B cells and we have the same constant region. Okay. 14. And if you guys don't have this, you guys can go to the Study Union, ucfstudyunion.wordpress.com, the biology tab. And if you guys came in late too and missed some of the ones going over, you guys can always come up here at the end and get the answers for it. Okay. So Cd4 cells are T helper cells, which include Th1 and Th2, and help induce an immune response associated with MHC class 2. That's true, good. CD8 T cells, or CD8 cells, are T cytotoxic cells that are associated with MHC class 1. Good. So a way that I like to remember these is you have the Cd4 cells, which go with MHC class 2. So you can think 2 times 4 is 8. And then we have CD8 cells that go with MHC class 1. So you can do 1 times 8 is 8. And so you can make sure that you have them correctly paired with each other. Okay, C. The adaptive immunity contains memory cells that allow the body to respond more quickly and vigorously to pathogens after 1 round of exposure. True or false? True, good. Okay, D. The adaptive immunity has tolerance, which means it has an acquired inability to make an immune response to certain antigens. True. What is the tolerance referring to? More so like, so we're not attacking our own cells. Mm-hmm, good. Okay, 15. Which of the following is true regarding Pmp interaction with PRR? A. PRM receptors are now called PRR and are found on the surface of B cells. Good. Why is this false? Okay. Okay. So the first part was actually true. PRM receptors are now called PRRs. But the second part's not true. They're not going to be on B cells. Okay, B. PAMP stands for pathogen-associated molecular proteins. False. What does it stand for? Good. Pathogen-associated molecular patterns. Be careful for stuff like that on the exam, because if you read it too fast, it sounds really right, and it's not. Okay. PRR stands for pattern recognition receptors that are phagocyte proteins bound to the membrane. True, good. So remember, the PAMP is going to be on what? Our cells or the pathogen? Good, the pathogen. They're pathogen-associated molecular patterns. And then all phagocytic cells have the pattern recognition receptors that bind to the PAMPs. Right, exactly. Okay, which of the following are false? PRRs were first observed in Drosophila and are called toll receptors. Good. So I got one answer for that, but that is true. B. Toll-like receptors on human phagocytes recognize specific PAMPSs. Good. C. Interaction of a PAMP with a phagocyte PRR triggers a transmembrane signal that results in transcription of a number of cell proteins in phagocytes, which lead to the production of toxic oxygen compounds that lead to pathogen death. That was a really long sentence, but yeah, that's true. D, the innate immunity includes the lymphocytes T cells and B cells. False. It's going to be adaptive. Good. Remember, like I said before, adaptive is just going to be T cells and B cells. Okay, 17. Which of the following is or are generated by the myeloid precursor? Monocytes which develop into macrophages. Good. Granulocytes. Okay. Neutrophils. Mast cells. Eosinophils. Basophils. Good, all of them. You're correct. Which one? She asked the question, the eosinophils bind with what antibody? Say it louder and prouder. Yes? Yeah? Did anyone hear? Yeah. Yeah. The IgE. And what is that called? Like, yeah, IgE is for allergies. What'd you say? IgE. Yeah, for IgE, I think of it like as the allergic reactions. It doesn't sound, it's not how allergic is spelled, but I think of it like that. So like allergic reactions. But don't do allergic because it's not right, it's not A. Okay, so all of these are from the myeloid precursor. We have granulocytes in here. Which of these are actually granulocytes? Good, so I think you guys said basophils, eosinophils, and neutrophils, right? So you can think the granulocytes are Ben, basophils, eosinophils, neutrophils. So if these all come from the myeloid precursor, what's the other precursor that we have? Good, the lymphoid precursor. And what cells come from that? Say it again? T cells, B cells, and say it again? And the natural killer cells, NK cells. Okay, 18. What is the name of the part of the antigen that is recognized by the antibody? Good, the epitope. Where is the paratope? So if the epitope is on the antigen, the paratope's going to be on the antibody. Great. I don't know if she told you guys paratope, but I just included it in there. Okay. Which of the following are true regarding immunoglobulins? What's another word for immunoglobulin? Good. The Igs or antibody. Okay. IgG has gamma heavy chains. Good. IgM has mu chains. True. 100% of the serum Igs are IgG proteins. False. How much is it? 80%. You guys are rocking it. Yes. Yes. So 20. The Pmp reaction is with IgM. False. Which one is it with? Cool. Okay, which of the following is correctly paired? IgG, first antibody to appear after immunization. Which one is that going to be? IgM? B, IgM, monomer on B cell surface receptor. It is true. So it's going to be a monomer when it's on the B cell surface receptor, and what is it in the secretions? Yeah, okay, so it has 5 of them. What's that called? Pentamer, good. Okay, and for each one that you have, so we know that it's a pentamer, so it has 5 of the individual little immunoglobulins. How many binding sites would it have? 10. Now, again, so if we're looking at this, how many heavy chains do we have on here? 2 heavy chains. So the heavy chains are going to be circled in red. So those are the heavy chains. And then these are going to be the light chains. So we have 2 heavy. And 2 light. Okay. IgA, dimer in blood and monomer in secretions. False, what's wrong with this? Good. Okay, IgD is involved in allergic reactions and parasite immunity. Which one is it? IgE. And then IgE, function unknown. Which one's that going to be? Yep, IgD. I think like IgD is like we don't know. Okay, which of the following is true? Select all that apply. IgG is the most common of the circulating antibodies and is composed of 4 polypeptide chains of 2 heavy and 2 light. Good? And that's what IgG looks like too, by the way. IgM, when it's in the secretions, it just has 5 of those. And what is linking all the chains together in IgM? What's it called? Okay, and more specifically a blank chain, a J chain, good. For IgM, okay. So IgG contains disulfide bonds. Yes, true. So disulfide bonds are gonna help to connect the heavy and light chains together and then help connect. Yeah, just A and M. And for A, when would it have the J chain? When it's in the blood or when it's in the secretions? In the secretions, good. Okay, IgM has 10 binding sites with low affinity and high avidity. Good. And IgA is a dimer linked by disulfide bridges to J-chain protein and is found in mucosal secretions of the gastrointestinal, respiratory, and genitourinary tracts. Sounded very uncertain, but it is true. So this one would not be true if it was not saying that it's in the secretions, because right now it's saying it's a dimer, but I said it's found in the secretions, so I'm giving you where it's found. So if it was in the serum, then it wouldn't be true, because what's it going to be instead? The monomer. Okay. Which of the following is false regarding MHC proteins? MHC stands for major histocompatibility complex. That's true. Good. T cells cannot recognize foreign antigens unless it is presented in the context of an MHC protein. That is true. So basically for T cells, they have to be presented with MHC. That's why we have the MHC class 1 and the MHC class 2. So we're going to have the MHC. If this is one of our cells, it's going to have the MHC. So let's say this is MHC. And then what goes next? Say it again. So we have the T cell receptor after that, and then we have the T cell. But what's connecting the two of them? The antigen, right? So this is one of our cells. Okay, MHC is a platform that bridges degraded antigens and presents them to the B cell receptor. Why is this false? Good. It's going to be for the T cell receptor. Huh? Oh, yes. So that's correct. That's the answer, because it's false. Yeah, that's another thing. Make sure you guys are reading the questions correctly and don't forget what they're asking for, because I know sometimes when I'm taking the exams and it's like, oh, which of the following is false, and I read the first one and I was like, that's true. And then I go ahead and bubble in my answer and then I move on, so don't do that. Okay, class 1 is found on the surface of all nucleated cells, and class 2 is found only on the surface of B lymphocytes, macrophages, and dendritic cells, which are in turn called all dedicated antigen presenting cells, or APCs. That's true. So all nucleated cells, so technically, could we have MHC class 1 on a B lymphocyte? Yes. Could we have it on a macrophage? What about a dendritic cell? Yes, right? What are the only types of cells that we are not going to see them on? Red blood cells? And there's one more. Starts with a P. Platelets. So those are going to be the ones that do not have any MHC. Yeah, no classes, because for MHC class 2, it's just going to be B lymphocytes, macrophages, and dendritic cells. And then for class 1, it would normally be like all of those included because they technically have a nucleus, but the only ones that don't have nuclei are going to be red blood cells and platelets. Okay, which of the following is used by a T helper cell to form pores that deliver toxic enzymes? A, granzymes. What do you guys think? Good, so it's not going to be that, but the granzyme is going to be involved in this process. How is the granzyme going to be involved in this? Good, so the granzyme is going to be what goes into the cell to help destroy it. The granzymes? Does anyone know the answer to that question? The question was, how do the granzymes help to destroy it? Yeah, with the granules, and they have basically like lytic properties. So it's going to go in and cause the cell to go through apoptosis and lyse, yeah. Okay, B granules. If you guys forgot what the question was. It's which of the following is used by a T helper cell to form pores that deliver toxic enzymes? Is it a granule? What about C, perforin? Good. So what is the perforin? Yeah, basically, so it's like forming a pore on the cell. So that the granzymes can enter. So the granules are just going to be like aspects that you can see under the microscope of the specific cell, yeah.
Like that one is like with neutrophils?
Yeah. So this question, we're doing time. So used by a T helper cell to help form pores that deliver toxic enzymes. So why do you think we would need to form a pore to deliver the toxic enzymes? What types of infection is this going to be for? Is it going to be like a bacteria that's extracellular, or is it going to be one that's intracellular? Good. This one's going to be one for like intracellular. So like maybe you have a virus that's infecting the inside of the cell. We need to find a way to go inside of the cell and destroy it. So they're going to form the pore with that's called a porphyrin, and the granzymes are going to go in and help to destroy the cell. Okay. Which of the following is true regarding NK cells? I'm going to put NK also stands for natural killer. Okay, so A, they are a class of lymphoid cells that instead of killing microbes, they destroy the host cells that harbor the microbes or have been transformed into cancer cells. Yep, that's going to be true. B, are unable to, that does not make sense. It should say are unable to kill virus-infected cells or tumor cells. Good, so that's false. Are unable to kill virus-infected cells. Okay, C, produce cytokines including TNF and granulocyte macrophage colony stimulating factor. Yeah, that's true. Okay, which of the following is true regarding DiGeorge's syndrome? It occurs in infants that lack a functioning thymus gland. That's true. So, okay, regarding this, what type of cells are going to mature in the thymus? T cells? And then where do the B cells mature? In the bone marrow, good. But where do all of the like immune cells kind of start? Besides for when they're like stem cells. A lot of them are going to be in the thymus, and then they can travel to the bone marrow of their B cells to mature there. So you have a lot of cells starting out in the thymus, and it's really important for your immune system. So regarding DiGeorge's syndrome, they're born without a functioning thymus gland, so they're very susceptible to getting disease and pathogens. So, okay. It causes increased rate of tumor production. True, right? Because if they have not as well of an immune system to respond to something, then they're more likely going to get tumors being produced and get a lot more diseases that way. So C, they are still able to reject transplanted tissue. Yeah.
Like for the last.
Does anyone know what TNF stands for? The tumor necrotizing factor. You want me to write it down? Okay. All right. So C, they are still able to reject transplanted tissue. So someone said false. That's true. Why is it false? Does anyone know? Okay, so basically they have no thymus, right? So they're lacking part of their immune response to foreign invaders. So if they're going to get a transplanted tissue, sometimes the transplanted tissues don't work, right? They're not compatible with the person that they put them in. But in this case, they're lacking the specific cells to recognize that. So they're not able to reject any transplanted tissue, basically, that they tried to give them. Okay, sterilization frees the treated object of which of the following? A, viruses. Good. B, endospores. C, all living organisms. Good. So it's going to be all of them. Okay, regarding ultraviolet light, which of the following is true? It leads to the production of cytosine dimers. Why is that false? Thymine dimers, good. It is most effective at 280. Why is it not 280? It's going to be 260. 280 is actually going to be for proteins. And 260 is going to be for DNA, which is why they can detect the thymine dimers there. And D, it leads to the production of tyrosine dimers. We just said it's thymine, so that's not correct. Ionization radiation has lower energy and longer wavelength. False, was it supposed to be? Good. High energy and short wavelength. Okay, which of the following is not correctly paired? Antimicrobial agent, a natural or synthetic chemical that kills or inhibits the growth of microorganisms. Good, that one's going to be true. B, disinfectants, chemicals that kill microorganisms and the spores. Why is this false? Good, so it's not necessarily spores. Sanitizers, less harsh than disinfectants and reduce microbial load to safe levels. Good. Antiseptics or germicides, kill or inhibit the growth of microorganisms and is non-toxic, which means it can be applied to human tissue. Good. Okay, which of the following is not correctly paired? Bacteriostatic, inhibits protein synthesis, and even if the agent concentration decreases, the cell is unable to grow. Why is this false? Exactly, good. So this one's going to be the correct answer because it does, in fact, inhibit protein synthesis, but if the agent concentration decreases, the cell is able to regrow again. The cells are able to regrow. Okay, so bactericidal, kills the cell but does not destroy the cells. This one's actually going to be true. So it's going to kill the cell but not necessarily destroy the cell. So the cell's technically like there, but it's not living, if that makes sense. It's not like the cell is like lysed. It's just like killing the cell internally so that it's still there but it's not completely destroyed. And then bacteriolytic cells, kills and destroys the cell, leading to the release of cellular content, as in lysis. True, good. Okay, which of the following is the tube dilution test looking for? A, zones of inhibition. Okay? What are zones of inhibition? Not quite, not quite. What's it called, though, for zones of inhibition? Not Kirby-Bauer. It starts with an A, but it's not technically that one. Agar. Agar zone. Agar zone diffusion. You guys got it. I believe in you. Great. Okay. 32. Bactericidal activity of a chemical compound in relation to phenol. No. What's this one going to be? Phenol. Phenol. Coefficient test. Thank you. For which one? For the phenol one? Does anyone want to help explain the phenol coefficient test for her? What's the main purpose of it? What is the purpose of even talking about phenol in there? Say it again? Say it? Okay. Basically, they're testing what you have basically, and they're testing it with one that's already known. So they're testing the compound that you have to see like what it's doing in comparison to one that's already known basically. They're just like, it's a comparative method to test if this one would be effective compared off of another one that they already have. Okay. C, minimum inhibitory concentration. Good, yay, right? So someone explain this, the tube dilution test.
So it's just trying to see like how much is required to be able to just like kill off the
Exactly. So you're looking, how are they doing this? So it's tube dilution. They're basically diluting it more and more, and then how do they know when they get to the minimum inhibitory concentration? One's cloudy and one's clear, right? So one's going to be clearing it, and then the next one is no longer clearing it. So they know that once it's no longer clearing it, they have passed the minimum that they need. Good. Okay, there you go. The Kirby-Bauer disc susceptibility test shows that larger zones of inhibition does not always mean more effective. That's true. Why? Yeah, exactly. So it could be depending, right? So it could be clearing. You could have more clearing, but what if the pathogen is going to be replicating at an extreme time? And so even though it's clearing it more, they could be replicating more, and it could be actually doing not as much as something that's not clearing as much, but the bacteria doesn't actually replicate for like 4 hours or something. Okay. 34. A bacteriostatic test only inhibits growth. Good, true. Okay, which of the following is not paired correctly? Antibiotics, chemical substance produced by one microorganism that kills or inhibits the growth of another. Good, that's true. B, synthetic antimicrobial agents created in hopes of finding a magic bullet. Good. C, semi-synthetic, modified antibiotic. Good. D, penicillin with modified R groups, synthetic antimicrobial agent. So this one's going to be false. It's not going to be synthetic. What is it going to be? Semi-synthetic. Good. Say that again. Synthetic antimicrobial. Oh, okay. Yeah. Yeah, it should be the antimicrobial agent. Yeah. No, sorry, antibiotic. I don't know why I just said that. Okay. Sulfanilamide, synthetic antimicrobial agent. And F, penicillin G, antibiotic. Good, that's true. Okay. Right, exactly, because they're modifying the R groups for it, so it's going to be semi-synthetic and antibiotic, because penicillin normally is antibiotic. Good call, Ryan. I didn't even realize that I had written that down. Okay, 36. Salvarsan was an arsenical agent effective in the treatment of herpes that was replaced by penicillin in the 1940s. False? Why is this false? Good. It's for syphilis. I don't know if I spelled it right, but it's okay. Okay, 37. Which of the following are correctly matched? Ehrlich and penicillin. Fleming and Salvarsan, and then Domag and Prontosol. C, what's up with A and B? They're flipped, good. Okay. Which of the following was the first commercially available antibiotic? Sulfanilamide, penicillin, salvarsan, and cephalosporin. What do you guys think? It's going to be sulfanilamide. But all you guys pick penicillin. I would go back and review those.
Isn't the sulfanilamide modified penicillin?
Okay, so the sulfanilamide, what do you guys know about that? Okay. What else do you know about it? Exactly. Exactly. So the sulfanilamide, looking at 39, sulfa drugs are also growth factor analogs. Is that true or false? And then we'll explain that. True. Okay. So what is the deal with these? Why are these created? Like what is the purpose of them? Why can they be beneficial? Okay, good. So they don't harm us. Why do they not harm us? Good. So basically, the sulfa drugs are going to be growth factor analogs of what? What molecule? It starts with a P. Good. Para-aminobenzoic acid, or known as like PABA. So what does this have to do, what does PABA do in bacteria? Before that. It's used to make folic acid. So the PABA is used to make folic acid, and then the folic acid can go and make nucleic acids. So this is beneficial to use against bacteria because the bacteria, this is what's happening in bacteria. They don't make folic acid on their own, they have to synthesize it. So if we are using a sulfa drug, like sulfanilamide, and we put it in here, it's actually going to be what type of competitor against PABA? Competitive, uncompetitive? Competitive. So it's going to be a competitive inhibitor of PABA. Do you guys remember those from the enzymes chapter? No? Okay. So sulfanilamide is going to be a competitive inhibitor of this, which basically means it's going to try to compete with PABA for the enzyme that makes it into folic acid. So it's competing with the enzyme that turns PABA into folic acid. So if it's competing with that, then folic acid's not going to be created, and we're not going to make nucleic acids. So because the bacteria is not going to be making nucleic acids, eventually the cell is going to die off because it's not going to have anything to survive. And this is only happening in bacteria because they have to synthesize this. This isn't going to work in eukaryotes because, or like us for example, because we actually take in folic acids in our diets. So what's that called when it works in one type of organism and not another? It starts with an S. It's selective, right? Okay, 40. Which of the following is not correctly matched? Penicillin G, broad spectrum with gram-positive and gram-negative. It's actually going to be false. Why is it false? So it's going to be narrow. And is it gram-positive or gram-negative? Good. So it's gram-positive only, so it's going to be narrow spectrum. So then ampicillin, broad spectrum with gram-positive and negative. This one's going to be true. And then vancomycin, narrow spectrum with only subset of gram-positives. I crossed this one out, but that's the answer because it says which one's not correctly paired. So this one's going to be true. Narrow spectrum with only subset of gram-positives. 41, fungi are prokaryotic. Good. False. Fungi are eukaryotic. What problem does that create for antifungal drugs? Exactly. So we also have eukaryotic cells. Fungi have eukaryotic cells, so they're not going to be as selective against one or the other. So they really have to find something that's specific to the type of fungi that's causing the infection, because otherwise they could actually go and inhibit our own cells, which is bad. Yes. Okay, so that was it for the first PowerPoint. Second PowerPoint. Which of the following represents the central dogma of molecular biology? Antisense strand, translation, mRNA, transcription, protein. False. Good. B. Template strand, transcription, mRNA, translation, protein. That's going to be true. And then C. Antisense strand, transcription, mRNA, translation, protein. True. So it's going to be both of them. So remember, you're going to have, you have your double-stranded DNA. Which one's going to be the template strand? The one that's 5 to 3 or 3 to 5? Three to 5, right? So this one's going to be the template strand, also known as the antisense strand. And then the top one's going to be known as the sense strand. And does anyone know why they use the 5 to 3' one as the template and antisense strand? Exactly. So when we synthesize the mRNA, we want the mRNA to be going 5' to 3'. And the only way that we can get it from going 5' to 3' is if we're looking at the 3' to 5' template strand. And okay, when you guys are looking at these strands, when they go from the DNA to mRNA, what is changing from the code? What's the difference in the nucleotides? Good, so the mRNA is going to have uracil. In replace of what? Good. So if she were to give you a question and gave you both strands of the DNA, and you were looking at the top one's complementary to the template strand, and the template strand's the one that we're making the mRNA from. So you can really just look at this top one and replace all the T's with U's, and it'll be what the mRNA strand would be like. And then if she just gave you the template strand, you would have to do the complementary version of it and replace all the T's with U's again. Okay. 43. Which of the following is false? Prokaryotes are polycistronic. True. And what does that mean that they're polycistronic? Good. They can code for multiple proteins. I'll say protein / polypeptide chains. Eukaryotes are monocistronic. Good, that is true. So the difference, one of the main differences is that prokaryotes, they can make multiple proteins at a time, whereas eukaryotes, we're just making one specific protein. So eukaryotes' DNA contains introns and exons. Good, that's true. And prokaryotes' DNA contains introns and exons. False. Why is this false? Which one don't they have? Good, they don't have the introns. So what is the difference between intron and exon? Do the introns code for anything? Good. They're non-coding, and then the exons are going to be the ones that actually code for proteins. Huh? The function of introns is you guys aren't going to get into it mainly in this class, but they can code for different like types of RNA and different like silencer RNAs and stuff that like affects translation in different aspects. But basically they're non-coding, so they're not going to turn into proteins. They can just help with processes that occur before translation, basically. Okay. So we just said that eukaryotes contain introns and exons, and we know that the introns are non-coding, so they're not going to help code for the protein. So we need to remove the introns prior to beginning translation because they're not coding for anything. So what is the name of the protein which removes the introns and joins the adjacent exons to form the mature RNA in eukaryotes? Good, it's going to be the spliceosome. Splicer gene, I just made up. DNA polymerase, what does DNA polymerase 3 do? Not opening it up. Kind of? Okay, it's for elongation, right? It's going to be adding in the nucleotides when they're being synthesized. So I'll say synthesis slash adds nucleotides. What about DNA ligase? Okay. Seals the gaps between what? Between what? Okay, the D, yes, good. The D, yes, the strands for the Okazaki fragments. Okazaki fragments. And where are those fragments going to be produced? Are those not going to be on the leading strand or the lagging strand? Okay, which of the following must occur prior to splicing? The addition of a methylated cytosine nucleotide in the 5' phosphate end. Nope. Addition of a methylated guanine nucleotide in the 3' phosphate end. Addition of a methylated guanine nucleotide in the 5' phosphate end. Good. So we have the 5' end. We're going to be adding the methylated guanine. And trimming the 3' end and adding a poly-A tail. Good. And trimming the 5' end, adding a poly-A tail. Right. That should technically be going over here, but I wrote the 3 before I wrote the A's. Okay, so C and D. Which of the following is true? Select all that apply. Cytosine and guanine make 2 hydrogen bonds together. Now, how many bonds do they make together? Good, 3. Thymine and adenine make 3 hydrogen bonds together. How many do they make? DNA is complementary and parallel in nature. False. So the first part is true, it is complementary, but it's not going to be parallel, it's going to be anti-parallel. Okay. Pyrimidines include cytosine and thymine. That's true. So we have purines. I don't know if this was in this chapter, but I know you guys definitely had this on the first exam. Do you remember what the purines are? Good. So adenine and guanine, you can think pure as gold. And then for the pyrimidines, which ones are left? And so for this one, you can think pure as gold and cut the pie. Cut pyrimidines, pure as gold. Which one has 2 rings in it? And how many rings does pyrimidine have? Good. Just in case you guys forgot from way back when. Okay, which of the following is true regarding the Missense and Stahl experiment? A, it showed that DNA replication is semi-conservative. Good, that's true. B, cells were first grown in all heavy DNA of 15 nitrogen. Okay. C, they use ultracentrifugation to perform this experiment. Good. Okay, so I'm going to talk you guys through real quick what the experiment did. So, what is ultracentrifugation? Good. So you're spinning basically the samples down, and how are they being separated? Good. By their weight and their densities, right? So what they did was they put all of this DNA into the first, they did all heavy. So they were in 15 nitrogen. And where was the DNA after this? Where did they find it? Towards the top of the tube or towards the bottom? Towards the bottom. And then what they did is they transferred it to light nitrogen, and they spun it down, and they allowed it to replicate. And where did they find the DNA? So they found it as a hybrid, half heavy and half light. This was after one round of replication. So then they kept it in the same light nitrogen for one more round of replication, so second round. And where did they find the DNA? It was in the middle, and it was up at the top. So what this showed was when they first did it, it was all at the bottom because it's all in heavy nitrogen. And then now it's going to be half heavy, half light. So it's going to be a hybrid in the middle. And then when they allowed it to replicate again, since it's semi-conservative, they're keeping half from what they previously had, and they're adding half of what's in there. So now they have half of the hybrid and half of only light because it's in the light one. So then if they were to redo the experiment, or if she were to ask you about this, and they were starting with light and going to heavy, where would they first start out? At the top or at the bottom? At the top. Where would it be after one round of replication? In the middle as a hybrid, right? Because now we're going from light to heavy. So we have half heavy, which is normally found at the bottom, and half light, which is found at the top. So that's why we see it as a hybrid. And then after the second round of replication, where would we see it? In the middle, and then down at the bottom. Cool. Okay, which of the following are not correctly prepared? Paired, not prepared. DNA polymerase 3, 5' to 3' synthesis. Is it true or false? True, good. DNA ligase seals the gap. RNA primer synthesized in 5' to 3' direction. Good. DNA polymerase 1 initiates DNA synthesis. Why is this false? Because why? Good. It should be DNA polymerase 3. And DNA helicase unwinds the DNA double helix. Good. All right. What is the function of the tau subunit? Remove supercoils. Good. So that's actually going to be gyrase, or we can say topoisomerase. Create supercoils. So, no. I mean, the supercoils are created, but it's not like, yeah, there's not like one specific job to make supercoils. That wouldn't help. Holds two DNA polymerase assemblies and helicase together. Good. So basically in replication, when you're making the replication fork, you know, you have your leading strand and the lagging strand, and you have the DNA polymerase. Let's say we have a DNA polymerase right here for the leading strand, and we have a DNA polymerase for the lagging strand. The tau subunit is basically going to like hold these two together with the helicase that is trying to separate them. Yes? Yeah? Say that again? What was the, okay, so what was the question? Like that's holding so the DNA polymerase 1 can come back in and fill in the gaps?
Yes, it stays.
Does anyone know that answer? I can't think of it off the top of my head. What is it? Oh, the single-stranded binding protein? Yes, there you go, the single-stranded binding protein. For when, he was saying for the lagging strand, when the DNA polymerase has left, and now we need DNA polymerase 3 is leaving, 1 needs to come in to finish sealing in the nucleotides, the single-stranded binding protein is going to bind to the single-stranded DNA to make sure that it's like kept down so that it can go to the next one. So that like the DNA polymerase 3 goes to the next primer, and so that it can continue. And that's another good question. I would check on that. But how many primers are you going to need for the leading strand? Good. What about for the lagging strand? Good. Multiple primers. Okay. Entire subunit adds the incoming nucleotides. No, we already talked about that. Okay, which sequence is known as the Pribno box? -35, -10, -5, -40. That's going to be -10. Is the Pribno box going to be in prokaryotes or eukaryotes? Prokaryotes. Does anyone know what the -10 sequence is called in eukaryotes? Yep. The Tata box. I like to remember that because like we're eukaryotic, we have Tatas. So, and then Pribno box, prokaryotes, P and P. Okay.
Like, I don't know, is that like part of
Oh, okay, so the replosome is basically showing you all of, it's like basically the term for like everything that's happening.
Oh.
So it's like showing you every single like piece that's in there. It's all part of the replosome. It's like all making up the replication, the replication machinery, if that makes sense. Okay, what do we have left? Which of the following represents the correct order of transcription? A, core enzyme and sigma bind. Sigma recognizes the promoter consensus. Transcription begins. Polymerase and RNA are released. Sigma is released. Why is this false? We'll go through the next ones. B, core enzyme and sigma bind. And what subunits make up the core enzyme? More specifically, like the Greek letters for the subunits in the core enzyme. Yes, so we have, well, kind of, it starts with alpha 2, beta beta, and then I think that's omega. And so then once we get the sigma to bind, then we would have sigma in here too. But this is the core enzyme without it. Okay, so core enzyme and sigma bind. Sigma recognizes the promoter consensus. Transcription begins. Sigma is released. Polymerase and RNA is released. Yep, that one's going to be true. So it's important that sigma is released prior to the polymerase and RNA being released. Okay. What causes chain termination in transcription? A, the poly A tail is recognized and cleaved, ending transcription. No, that one's not true. B, a run of uracils is used to cleave the poly A tail. What do you think? No, that's not true. C, inverted repeats hydrogen bond together in a stem loop structure in RNA. Good, okay. So basically, once you're going through transcription, it's going to be going, and then all of a sudden it's going to form the inverted hair loop. And then you have the run of uracils that are down here at the end of the hair loop. And also important to note that this hair loop forms because these sequences right here, these nucleotide bases, can base pair with each other. So you're going to have base pairs that are complementary, and then some in here that are not. So if she were to ask you, like, which of the following could form the stem loop, you have to have starting ones. You would have to have like A here and U over here, so these could bind, and then maybe like C and G, so those could bind, and then we could do like G and C, and then those would make up going over here. So if we wrote it like this, it would be A, C, G, and then let's say we'll just do like A or something. Then A, and then C, G, U, so that these bind together, and this is in the part that's not binding. Does that make sense to everyone? Okay. And is this going to be row dependent or row independent? Row independent, good. And I don't believe that she even gave you guys a slide on what row dependent looks like, so keep that in mind if she tries to trick you. You guys didn't even have that in your PowerPoint slides. Just saying. Because it might sound really good on the exam, but you guys didn't even learn it, so they're not going to test you on that. Okay, the sequence reaches one of the stop codons. Where are the stop codons? Is this going to be in transcription? Good, so this is how this would stop translation. Okay, 53. A missense mutation creates a stop codon. False. What does a missense mutation create? Good. It's just going to form, like, it's going to create a different amino acid, but it's not a stop codon. Well, technically, since it's coding for an amino acid, it's not going to be the stop codon. The stop codon, its tRNA molecule actually has nothing attached to it. So once you get to the stop codon, there's going to be a tRNA molecule that binds that specifically has no amino acid. So this one has to make something so a new amino acid could attach to it. Yep, good. So yeah, the nonsense is what creates the stop codon. And you can think nonsense like it's not sensing, it has no sense there, like you can think like it's nonsense to stop early, I don't know. something like that. So 54, a silent mutation does not alter the protein. That's true. How could the silent mutation not alter the protein? Which part of the codon do you think it would have to be? Which nucleotide? So you're getting some mixed answers. So it's actually going to be the third one. Do you guys remember in the PowerPoints where they're saying that the genetic code is degenerate? No? Got one head nod, and then she switched to no? Yes? Okay. So basically, degeneracy. There's degeneracy in the codons, basically meaning there's going to be 3 nucleotides in the codon. So let's say like, I don't know, A, G, C. So this third one, it's kind of like, it's also called the Rabbi Hypothesis. I don't know if she talked to you guys about this, but the reason why you can create a silent mutation is because this third one's not really as critical to the amino acid as the first one. So if you guys remember looking on the chart that she gave you guys, and it would like show you all the different pairs that you guys could make for the amino acids, and there was like 4 for each one, and some of them had like 2 or 3 for each one. If you guys notice, the first 2 letters are usually the same for all 4. It's just going to be the last nucleotide that's different. And basically it's just this last nucleotide, multiple of the codons can code for the same amino acid. So you can switch it, and if we were to switch this to, let's say, like G or something, I don't know if this is like real because I just made these up right now, but this could technically code for the same amino acid because it's switching it in the third pair. But if you were to switch it in the second pair, that's when you're more so going to make either the missense mutation or the nonsense mutation. Yeah, in the third position, like the one that's going right here. Okay, so what are the 3 stop codons? I don't think you guys, I think she told you guys you didn't need to memorize that, but it's A, just in case. And they were up here, just 2. Okay, what form of genetic material do retroviruses contain? Single-stranded DNA, single-stranded RNA, double-stranded DNA, double-stranded RNA. Good, it is single-stranded RNA. And what happens when the retrovirus actually infects the animal cells? Are they going to be coded or not coded? So first they're going to be coded, and then once they enter inside, then they're going to become uncoded. Okay. Wow, okay, so yeah, it's basically talking in the next question. What correctly depicts the steps in the replication of retroviruses? So entrance, reverse transcription, uncoding, travel to the nucleus, transcription, integration into host DNA. Good, that one's false. B, entrance, uncoding, travel to nucleus, reverse transcription, integration into host DNA, transcription. What do you think? No, when we get to that one, we'll chat it out. Okay, so C, entrance, uncoding, reverse transcription, travel to the nucleus, integration into host DNA, transcription. Good, that one's true. So what is reverse transcription? And why do the retroviruses need to go through reverse transcription? Say that again? Yeah, exactly. So when they come in, right, they're in single-stranded RNA, and they need to use our host's machinery, right? The viruses can't do anything on their own. They need to be inside the host cell so that they can use the host cell stuff. So when they enter into the host cell, they're in single-stranded RNA, so they just have one strand of RNA. But we need it to be back into DNA so that it can use our polymerases so that it can be transcribed, right? And continue to be translated also so that it can make more of the virus. So it starts off with single-stranded RNA, and it's going to come in with the coat around it. So right when it gets in there, it needs to uncoat, right? Because if it's not going to uncoat, then how are you going to get the virus out? So it's going to uncoat. It's going to reverse transcribe. So it's going to go from single-stranded RNA back to DNA. Yes, exactly. And then it has to travel back to the nucleus. Why does it have to travel back to the nucleus? Right, because that's where transcription occurs, right? So it's going to travel back to the nucleus, integrate into the host DNA, because now we have this DNA that can be integrated into the host DNA. It's no longer single-stranded, and then it can be transcribed. And it's going to be transcribed into our RNA, into our, not our RNA, so that it can continue. So what's different about?
Like it doesn't have to like go back.
So what's different about it?
Like it doesn't have to go back to
Yeah, but why?
DNA. It's already
Yeah, exactly. So if it's coming in DNA, it's not going to have to go back and reverse transcribe, because it can just go ahead from there. So the only reason why this one has to is because it needs to be replicating. Like we need it to replicate. Well, not us, but the virus needs to replicate so that our cells like don't really like see it as foreign, and so that it can use our machinery. So if it comes in as RNA, it needs to be like reverse transcribed, but if it comes in DNA, it can kind of like skip over that. Okay. Well, normally we would not reverse transcribe something, but the virus itself is going to reverse transcribe so that it's like tricking our cells into reverse transcribing it. Yeah. And it's doing this so that it can go forward and use our polymerases and all of our machinery so that it can continuously replicate, because it's going to be integrated into the host DNA. That's what it's trying to do. It's trying to like integrate into our host DNA so that we don't really like recognize it's a problem until like there's tons of it. Yeah. Okay. You're welcome. Okay, so another experiment that I want to talk to you guys about real quick is the Hershey and Chase one. I remember that was also in your PowerPoint slides. Does anyone know anything about that? That one's a pretty good one. What were they using in this? Okay, so they're using bacteriophages, good. What is a bacteriophage? Okay, and what's inside of it, DNA or RNA, or both? Or? So it could be DNA. Yeah. It could also be RNA, right? And if it's both, what's it called? Mimivirus. So it could be DNA or RNA, and then the mimivirus is both. And what was the purpose of this experiment? What were they trying to find? Say it again? Yes, exactly. They're trying to see what was in the virus, right? So what did they label? They labeled two sets of bacteriophages with labeled what? What did they use? Sulfur and phosphorus. Why did they pick sulfur and phosphorus? Which one was for which? Which one was for protein? Which one was for DNA or RNA? Sulfur is for protein, and the phosphorus was for either DNA or RNA, right? Because there's going to be sulfur in the proteins, in some of the proteins we have, and there's going to be phosphorus in the DNA and RNA with like the triphosphates. So they basically labeled the bacteriophage, and they allowed it to go into the cell and infect the cell, right? And then how did they determine that it was which one? Which one did they determine it was? It was going to be DNA, right? Because all the phosphorus was contained where? Where did they find all the phosphorus? All the labeled phosphorus? Yeah, inside the cell, right? So they found this inside the cell that they infected. And where did they find the sulfur at? outside the cell. So because they found the sulfur outside the cell, and because they found the phosphorus inside the cell, they knew that what was infecting the cell had to be either DNA or RNA. Okay, and another one from the first PowerPoint slide is she talked about hapten. What is hapten? So it's going to be, you're correct. You're correct on that. So depending on if it's going to be by itself or if it's with a high molecular weight protein. And before you guys leave, by the way, there are like little surveys at the end of the table. If you guys could fill them out, it's super easy. You just have to like go down the line and write like strongly agree for everything. And then just put them back in at the side of the table. But so basically hapten is a low molecular weight antigen. And what happens when hapten is inside the host by itself? Does it create an immune response? Say that again? No, right? So if it's by itself, it's non-immunogenic. And what does it mean if you're immunogenic? Yeah, you get an immune response. So this was the purpose of her including this in the slides, was to show you guys, that not all antigens are also immunogens. Because when hapten enters, if it's by itself as a low molecular weight antigen, you're not going to have an immune response. So it's basically like if this pen was hapten and this room basically was the host, and it entered as itself and it's just sitting here, and the host, this room is like, okay, what is that? We don't really care about it. It's tiny. It's like minuscule, right? But now I'm standing up here. I'm a high molecular weight protein that's in the system, and now I'm holding this, and they're like, what is that? Get rid of that. Like, who is she, basically? So when it's by itself, it's not immunogenic, but it can attach to high molecular weight particles inside the host, and at that point, then the immune system is going to go and try to attack it. Yeah, not by itself, but if it's attached to a high molecular weight. So that's like the whole purpose of it. So it is an antigen, but it's only going to be an immunogen and cause harm if it's attached to something. Now, what happens is after it attaches to the high molecular weight protein, we're going to stimulate the T cells and B cells to go and try to fight it, right? So then once the infection is cleared, if hapten enters again, just as the low molecular weight protein, so like just as the pen in this case, the T cells and B cells that have formed memory cells are going to recognize that, and then they will go and fight it, and you will have an immune response like immediately. It doesn't need to attach to a high molecular weight because it's already seen it now and it knows that it's foreign. It's like, you can't trick us anymore, and it's going to go and make an immune response. Okay, so we have like 20 minutes left. We can talk about whatever kind of questions you guys have. What would you guys like to go over? Yes?
What does the IL stand for? Like, for the class 1? No, class 2.
Interleukin.
Because I know she, like, mentioned it at one point, like, for both of them. Like, the IL-1, like, 4, 5, and 6 for Th1.
Mm-hmm.
And then IL-2 is for the TC.
Mm-hmm. Yeah.
Right?
So the interleukins are basically going to be like, does anyone know the answer for that? What the interleukins do? Say that again? Yes, kind of. So, anyone have another response to that? The interleukins specifically? Huh? They can help to create buzz. So basically the interleukins are going to be either associated with your MHC class 1 or MHC class 2, depending on which one is which, and they're just going to go and help carry out basic immune responses. For that, I would just like basically know which ones go with which, because I think that's like all she really had in there, unless she went into specific details on it.
It was 4, 5, and 6.
Mm-hmm.
TH.
You said it right the first time. Yeah.
Oh,
Okay. So, all right, let's cover that real quick. So, MHC class 1 and MHC class 2. Why are they different? Okay, that was louder. Okay, one presents a CD4, one presents a CD8. Which one's CD4? Okay. And then this one's CD8. What types of cells are CD8 cells? TC cytotoxic cells. What type of pathogens are these going to try to fight? Those that go inside the cell, right? So intracellular pathogens. And what's a common intracellular pathogen? Viruses? Good. So CD4, what are the type of cells that are classified as CD4 cells? Good, so we have the T helper cells, TH1 and TH2. Now, which one is the inflammatory one? TH, which one? You got? Okay, we're having a consensus. Which one thinks it's 1? Which one thinks 2? Okay, it's actually going to be 1. And what are they going to be releasing? Good. Cytokines? What are cytokines? Kind of. So cytokines are basically releasing some sort of like signal, basically telling other things to do their job, basically. So they're going to release cytokines that are going to go and cause inflammation and a bunch of other things at the site of infection, whereas TH2 are going to do what? Good, they're going to activate the B cells. They're going to activate the B cells to turn into what type of cells? Good, so plasma, which secrete the antibodies. And then after the infection's done, what can the plasma cell turn into? Memory cells. Okay, so quick question. The T cells and B cells that you guys have that are released for the specific infection, are they all going to die super fast? Are the majority of them going to die super fast? Yes, right? So once you basically have the T cells and B cells that are specific to that pathogen, they're going to replicate like crazy. So like I said before, like if we were all B cells or we were all T cells and one pathogen comes in here and I'm like, that's me, it's mine, like this is my job. I'm going to go over there and I'm going to make like however many of me I need to secrete enough antibodies and to make enough T cells basically so that we can clear the pathogen. But then after we're done, we have all of these antibodies and all of these T cells that are sitting there and we don't need them anymore and they're just causing inflammation and a bunch of stuff that we don't, like the pathogen's gone and we're still inflamed. So they're all going to die off so we can try to get rid of that response. But we're going to keep a few of each just in case we get infected again by it. Thank you, good luck. Does that make sense? Okay. What else? No one has any questions they want to cover?
There's like a specific slide that like I'm confused by just like the process of like keeping them.
Sure. What's your question?
The activated B cells, they turn to plasma cells, and then the plasma cells will turn to memory cells, or do the activated B cells turn to plasma cells and memory cells? Because I didn't know that memory cells came from plasma cells. I thought they
So basically, I just wanted to emphasize that, like, you're not going to get memory cells until you have something that's been secreted in the antibody. Like, you want to make a memory cell out of the cell that's specific for that. So you're not going to make memory cell out of, like, not an activated B cell. I was just kind of more so like showing that the activated B cell gets turned into a plasma cell which secretes antibodies, and then the memory cell is specific to that one, if that makes sense.
But the plasma cell's not turning into a memory cell.
Right, it's just that specific B cell they're going to keep as memory cell. Yeah. Because the plasma cells are like when it's secreting the antibodies. Yep.
The antibodies directly kill the pathogen?
Say that again?
Do the antibodies directly kill the pathogen?
Mm-mm.
Okay. Because I mean, I figured out what
Mm-mm. They're non-phagocytic.
Okay.
Okay. What is this? PRM.
They just secrete the stuff that needs to be
Yes, pretty much. Yeah. They're going to help, like, once you get antigen interacting with antibody, then you basically, body is known, like, we need to do something and get rid of this. So it's going to call in all the other players and try to get rid of it. Okay, so she wanted to go over this slide. This is in the PowerPoints for you guys. It's basically talking about the Pmp interacting with PRRs. If you guys, I'm going to zoom in so we can kind of, like, discuss this. Never mind, because you can't read it at all. Okay, so, yeah, no. Let me just try to draw it out for you. It is kind of difficult. So this is basically showing you, this is drawn. We have your cell here, and this is your phagocyte. Okay. So basically, we have these PRMs, the PRMs and the PRRs, right? Associating with the pathogen. So let's assume I'm going to draw the pathogen in green. So the pathogen comes in. Here's your pathogen. The pathogen has the pathogen-associated molecular patterns on them, right? So basically something that's like sticking out of the pathogen that we can recognize. We have the pathogen, the pattern recognition receptor on our phagocytes. So basically, it can come in and bind to our PRR. So this is going to be the PAMP. This would be our phagocytic cells PRR. So once the pathogen comes in, it can bind to our phagocyte, right? And then what this picture is trying to show you is they have one for innate, and then they have one for adaptive. So if they're using innate, basically what's happening is we're releasing cytokines and chemokines, causing inflammation, trying to kill the cell. So this is all happening in innate. If you guys see the picture again, this whole first part, this is what's happening in the innate process. But this is also showing you that on the same cell, we have MHC molecules, this being the MHC, okay? So let's say we have the pathogen, thank you, and we can't clear the infection. It's been like going on now for a few days and we can't clear it. What's going to happen is this phagocyte's going to try to bring in this pathogen for destruction. Okay? It's going to bring it in, and the destructive pathogen, part of the destructive pathogen is now going to go and they're going to put it on the MHC class molecule. So the MHC class molecule now has part of the pathogen on it, and it can then go and bind to a T cell. So it's going to go and find the T cell receptor, this would be the T cell receptor, on a specific T cell. So now what happens is you have a T cell with this antigen basically bound to its T cell receptor. So what's going to happen now that we're in this part of the picture? So we're at the T cell with the activated, we're in the activated T cell with the antigen on it. Then we can go and also cause inflammation and do the same processes over again, trying to kill the cell more. And also now, we can activate the B cells. Since we have a T cell that's activated now, thank you guys, we can go and activate B cells, which are going to secrete the antibodies, which can go and bind to the antigen and try to destroy it. So basically this is showing you that once you have the Pmp interacting with the PRR, it's going to try to carry out the immune response first. It's going to try to kill it on its own. But if it can't, then we need to call in the adaptive that's specific to that pathogen, so that we can call in the specific T cells that can go and cause inflammation and release cytokines to help with that, and they can also go and stimulate the B cells that are specific for that type of antigen that can go and bind their antibody to the antigen, which kind of like neutralizes it, and it's like, okay, we're good. It's like, I try to give this analogy in Sark, but no one understood it. But when you guys were young, did you ever play that like Sharks and Minnows game where you're at one side and you're trying to get to the other side, and once they tag you, you have to stop? That's basically like antigen binding to antibody. So you're like running around and you're like, yeah, I'm going, and then they tag you and you have to like stop there. That's basically what's happening when your antibody is binding to the antigen. It's like tagging it, and now it's like neutral, and it's like, oh, okay. And then they can bring in like a phagocytic cell and like engulf it. Does that make more sense? Yeah. Th2 is going to stimulate the B cell. And then if it's the cytotoxic T cell, that's when we said that we're going to have them to go and try to kill it with other ways, one of them being forming the perforin pore and putting the granzymes in so that the granzymes can go in and cause the cell to lyse. The cytotoxic. Yeah, in cytotoxic T cells, you can think TC or cytotoxic T cells or CD8. And also, just make sure you guys don't get confused that CD4 is Th1 and Th2. It's the term for Th1 and Th2, and both of these combine, Th1 and Th2. Those are going to be called the T helper cells. But yeah, what are the three cells that are phagocytic? Monocytes, macrophages, and neutrophils. Good. So before the question with if the antibodies directly go and engulf the pathogen, they're not going to be phagocytic cells, so they can't do that. They're just kind of like making it neutral so that other people can come in and destroy it. And like we said before, with the MHC, we know that MHC can be on all nucleated cells if it's class 1, and only the antigen-presenting cells if it's class 2. So technically, in this phagocyte, we could have MHC class 1, and we can also have MHC class 2. And it's going to know which one it needs to be presenting it to, depending on if the pathogen is intracellular or not. If it's intracellular, it's going to go to MHC class 1, and if not, then they're going to put it on MHC class 2. And they're basically doing that so our cells can recognize which MHC it is, so that different T cells can bind, and depending on which T cells bind, that's going to create the different responses for the specific pathogen. Yes, MHC1, these are going to be all nucleated cells. Whereas MHC2 is only going to be B lymphocytes. What are the other two? Okay. And macrophages. So technically, like I said, the macrophages could have MHC class 1 and MHC class 2. And we need the MHC molecules because that's how the T cell receptor on the T cell is able to recognize the antigen. That's why they're called like antigen presenting cells, because they're physically presenting the antigen. They're like, here it is, come and get it type deal. Yes? Yes. The only ones that would have MHC class 1 and 2 would be B lymphocytes, macrophages, and dendritic. These are the only one that would have both of them. So don't let her confuse you on that one. I say her, but them. Huh? Yes. These ones can have both because these are technically nucleated cells as well. And also, if you guys are like done with the final and you find that you guys have nothing to do for the start of your break, you guys can go to the SARC website. It's like you type in UCF SARC under Google, you go there, and you click on tutoring, and then you click on rate my tutor, and you say really nice things about me. Okay? And my name's Angela, just in case you guys forgot. I need like 10 of them per semester, so it's a good way for you guys to do that. But any last minute questions? We have like 5 minutes left. This one? Mm-hmm. Thank you. Yes. Where is that?
And what is it constantly looking for?
What is immunosurveillance? What is it constantly looking for? Okay, so the question was, what is immunosurveillance and what is it constantly looking for? Who knows the answer to that? Kind of, yeah. So it could be looking, it's like basically they're like patrolling. So they're looking for antigens and they're making sure that they don't attack our own cells. They, like the immune cells.
Okay.
Like specifically, I would say the innate ones because the adaptive aren't going to be turned on unless they like need to. So ideally, the adaptive wouldn't come on for a few days, and it's only going to be if they can't clear the pathogen. Yes. Yeah. Yes. Good question. Yes.
Because you said that bactericidal like only like kills the inside. That's why like you'll still be able to see.
Yes. So good call. So when we were talking about this earlier about bactericidal and bacteriostatic, you have really pretty handwriting. Anyways, so right here you guys can see for bactericidal, they're killing the cell, but they're not necessarily destroying the cell. So that's why right here is when they add the arrow is when they add the agent. So they're adding the bactericidal right here at the arrow. The viable cell count goes down, meaning the living cells are like decreasing, going down the toilet because they're dying, and your total cell count is staying constant because they're not being destroyed. So it's like if all of us were in here and then we all died, but like no one took us out, if that makes sense. So we're all dying, but our total cell count is staying the same because we're still chilling in the room. Whereas for bacteriostatic, this is saying it's inhibiting the growth, right? So it's not necessarily, it's just inhibiting the growth. That's why we have the viable cell count and the total cell count are still going to be the same. And then down here for bacteriolytic, that's actually causing lysis in the cell, and they're being destroyed when you lyse them, right? They're like cutting the cell open, everything's like spilling out. So you have viable cell count going down and total cell count going down because they're dying and they're being destroyed. Good question. Did that help? Cool. Thank you. Yeah, and if anyone missed the beginning, if you guys want to come up and take pictures of the answers, that's fine too. And good luck on your exam. You guys are all going to do great. Thank you for coming. Yeah, go ahead.
I have one more question. For the test in the morning, and I'm really stressed.
No, you're going to do great. You're answering all the questions.
That was me, like,